Right. Anybody watch Whose Line Is It Anyway? All right. Anyway, it's one of my I used to enjoy watching it. Uh, but basically, it's whose genome is it anyway? So they decided that the genome that they were sequenced is not going to be any particular individuals. They actually mix the DNA, mix a race. So it says here, back libraries provided the DNA templates for human genome sequence in the public funded Human Genome Project. And for ethical reason, it was decided that the cells used to provide the source DNA would come from multiple individuals who would be guaranteed anonymity. Okay, so it's not any particular individual. Okay, the following section talks about genome projects for a diversity of model organisms. So they started off with E. coli, the bacterial organism, Saccharomyces cerevisiae, the yeast C. elegans, the nematode, and then Drosophila melanogaster, and then of course the mouse genome. All right, and then they found that having the mouse genome really helped because I've noted down at the book is called what they call as syntenic regions. Syntenic means regions that are almost similar in sequence and organization, right? There are certain regions. They may not be at the same place. For example, I can't remember on top of my head, but maybe chromosome 5 in humans is the same as chromosome 9 in mouse. This is just an example. I'm not sure whether that's even correct. But and then when they looked at this thing, they say that the order of the gene is the same. The sequence may not be entirely similar, but the genome, the gene, the order of the gene, the how it is being organized is almost the same. Certain regions, so these are called syntenic. Okay, finally, in the next section, it talks about powerful genome databases. So genome databases are not just repositories, they're not just databases where you can retrieve sequences. They also give you a view of the genome, and newer databases allows you to even do analysis. Browsers help to store and analyze. That's the important thing, right? And analyze. So from the earlier stages of the human genome analysis, central computer repositories were established for storing mapping data and sequence data produced in the laboratories. An important advance was the development of genome browsers with graphical user interface GUI. Why? Because when a lot of people, when they do bioinformatics analysis, when they do all this um, bioinformatics analysis, they like to use commands. They like to use like Python or scripting and so. Why? Because dealing with graphics takes a lot more time and computing power. So normally, because you have the sequence, they are text, so it's easier for them to actually just play around with sequence, and only later, once you want to share your information, you put it up in a graphic user interface. Graphic user interface is the what you see on the World Wide Web, the internet. This is a graphic user interface. But if you do analysis, even now when you do analysis, let's say for example you're interested in bioinformatics and you want to do genome analysis, you'll find that more often than not you'll have to rely on programming. Okay, on to the next video.